everyone, and welcome back to Byhollyg. Welcome to today's video. So this one is going to be all about bacteria, the biology of bacteria, and I am planning some videos where I talk about like antibiotic resistance and the gut microbiome and the microbiota. So I thought it would be useful to film this video first so you at least know what bacteria are, what their structure is, just a bit about bacteria. It's going to be a crash course on the biology of bacteria, and if you are looking for a video that you can use to kind of like help alongside this one, I recommend the tour of an animal cell, because you will then be able to compare a bacterial cell with an animal cell. So I hope you enjoy this video. Anyway, as usual, definitely give it a thumbs up if you do, comment down below, subscribe if you are new, and hit the bell so you know when I upload. And yeah, we will dive straight into the video. The first question then is, what are bacteria? So we need to firstly get to know what they are, and bacteria are what we call microorganisms, or microbes, the shortened version, meaning that they are microscopic. We cannot see them with the naked eye, and we have to visualize them under the microscope. They are single-celled microorganisms, but sometimes they will combine temporarily or permanently to form these colonies. Bacteria are also everywhere, like on our bodies, in our bodies. They can live in really extreme environments where most other organisms don't live. They can therefore be extremophiles or extramophiles, for example living in volcanic craters, so really hot and harsh environments. And what's really quite important here is that we have three domains of life. We have bacteria, archaea, and eukaryotes. And the bacteria and the archaea, they are what we call prokaryotes, in contrast to eukaryotes, which includes animals, so ourselves, plants, fungi, and others. And the defining difference between a prokaryote and a eukaryote is that prokaryotes, so bacteria, they do not have a membrane-bound nucleus. So their genetic material, their DNA, is not enclosed in a membrane, because in eukaryotes our DNA is found in the nucleus, and the nucleus is a membrane-bound organelle. And an extension of that is in eukaryotes we also have other membrane-bound organelles. So alongside the nucleus we have like the Golgi, the endoplasmic reticulum, we also have the mitochondria. A lot of the organelles in eukaryotes are therefore membrane-bound, and you'll know that if you watch the video looking at an animal cell in detail. There are other differences between prokaryotes and eukaryotes, and I will touch on some of them a bit later. One obvious one is that bacterial cells and prokaryotes are smaller than eukaryotic cells, but that is just the defining difference that is really important to know, is the presence or absence of a membrane-bound nucleus determines whether it's a prokaryote or a eukaryote. Question number 2 is, what is the genetic material of bacteria? And as I've already said, they have DNA, but it's not found in a nucleus. So bacterial cells, they have a main chromosome, and that is a large circular chromosome that carries essential genes, so genes that are needed for the survival and the reproduction of those bacterial cells. If you took away those genes and that main circular chromosome, the bacterial cell would not be able to survive. And because that's not membrane bound, we say that it is found in a space in the cell, in the bacterial cell, called the nucleoid. And sometimes the term nucleoid will be used to define the chromosome itself, so the DNA, in complex with DNA binding proteins. The term nucleoid is just used in different ways. But additionally, on top of that, as well as the main circular chromosome in bacterial cells, we also have these things called plasmids. And plasmids are small circular molecules of DNA. They are kind of extra chromosomal molecules of DNA because they are separate and distinct from that main bacterial chromosome. And they're really interesting because they are mobile between bacterial cells, which I will talk a bit about later, but they carry non-essential genes. So if you took away the plasmids, the bacterial cell would still be able to survive, because what they provide is kind of like additional or accessory genes. We can say that it's an accessory part of the bacterial cell genome because it allows bacterial cells to